This series of video will now talk about indicators and buffers. As you recall, indicators we discussed in Chem 20, but buffer is a slightly new topic for Chemistry 30. As you recall from earlier courses, an acid-base indicator is really just a conjugate acid-base pair. In fact, it is a weak conjugate acid-base pair. Now, ideally, at least one or perhaps both of these entities will be colored. When the indicator is acting as an acid, we call it HIN, and we call this the protonated form. When it acts as a base, it loses its proton. So now the proton is free-floating, and the indicator is now IN minus. We call this the basic form, the deprotonated form. And to act as an acid-base indicator, the protonated form must have a different color than the deprotonated form. A very common indicator we use in the lab is phenolphthalein. Now, when it's in its protonated form, so HPH, it is colorless. However, when it is basic or deprotonated form, it is now pink. This reaction, the protonation or the deprotonation, happens over a very narrow pH and happens very quickly. Therefore, it is a good indicator for use when we're doing acid-base titrations. Most indicators are complex organic molecules that have various aromatic rings, such as these, benzene rings, and it is these aromatic rings that give the compound some color. The protonated form has one extra H, whereas the deprotonated form does not have the hydrogen. In the case of phenolphthalein, this also causes a single bond oxygen here to be converted into a double bond, which affects the bonding of the entire molecule. That's why this changes color such drastically. Because these organic molecules are so complex to write out, we usually just indicate it as an abbreviation. So the protonated form of phenolphthalein, we just abbreviate as HPH, and the deprotonated form as pH minus. Let's look closely at another indicator, bromothymol blue. We'll abbreviate it as HBB for the protonated form and BB minus for the deprotonated form. We know that the Ka for this is 5.0 times 10 to the minus 8, which means that at equilibrium, at the center of the indicated range, the equilibrium expression is hydronium times the concentration of BB minus divided by the concentration of HBB. However, in the middle range, the concentration of the protonated form and the deprotonated form is the same. Therefore, they cancel out. Therefore, the Ka is really just the concentration of hydronium at the middle of the range. So we can actually calculate the pH of a solution if bromothymol blue is just in the middle of the range, and we can calculate the pH to be 7.3. We can link the behavior of acid-base indicators back to Le Chatelet's principle. Here we have a general equation for all indicators, and we can see that if we add more acid here, because we're adding more products, we will shift the reaction in the reverse direction backwards to form more products. In this case, you'll get more of the protonated form of the indicator. In this case, this is the litmus indicator, and we know that in acidic solution, when there is an excess of hydronium, the litmus paper will turn red because the reaction is shifted to the reactants. We can also think about what happens when we add additional OH ions. The OH ions remove hydronium. If we remove a product, then the system alleviates the stress by shifting reaction forward. We're going to favor the products. Therefore, there will be additional deprotonated indicators, which is blue. The pH range at which the indicator changes color depends on the strength of an acid. Remember, indicators are all weak acids and weak bases. However, they vary in their degree of weaknesses or their strength. These values have been measured and are on page 10 of your data booklet. Now, because these Kas have been measured, you could actually include them in your table of weak acid and weak bases. However, we rarely do that because we use indicators in such small amounts. They will not react in any major way with our reagents. The next topic we need to discuss is a buffer. In real life, the term buffer means ability to absorb some changes without major impact to your system. In acid-base chemistry, this is also true. It will able to absorb some amounts of a strong acid or a strong base without changing the pH of the solution. Now, what a buffer is, it is a conjugate acid-base pair. In fact, it is a conjugate weak acid, weak base pair that maintains a constant pH when small amounts of a strong acid or base is present. This has to be the same conjugate acid and the base. And here it refers to it as a salt. However, it's not table salt, it is just the weak base with its counter ion. In this case, in most cases actually, it is going to be sodium. So an example of a buffer system is methanoic acid along with sodium methanoate. A non-example of a buffer system, so a non-example, would be, let's say, again, methanoic acid and, let's say, acetic or sodium acetate. These are not the same conjugate acid and bases. Therefore, this is not an example of a conjugate acid-base pair or not an example of a buffer. Okay. As well, buffer solutions have a specific pH due to the nature and how much of those conjugate acid and bases are present. You will find that if we have a buffer system with methanoic acid, it will be different than a buffer system we establish using acetic acid. Let's take a closer look to see how exactly a buffer works. First, we're going to write the acid expression, the Ka expression. So here we're going to have acetic acid or ethanoic acid. It reacts with water to produce the acetate ion and hydronium. And this is going to be a reversible reaction. You can have the forward reaction or the reverse reaction. If we add additional amounts of hydronium in the form of, let's say, HCl, we're going to increase the concentration of the product. Therefore, we're going to shift the reaction in the reverse direction. We will produce additional amounts of acetic acid. However, acetic acid is not a strong acid. It does not ionize 100%. Therefore, we're going to soak up or remove hydronium. And if there is no extra hydronium, it does not change the pH of the solution. Conversely, if we're to add OH ions or add hydroxide ions, we will now shift the reaction to the right. The hydroxide ions will react with the hydronium to produce water, and because water is a pure liquid, we do not include it in our equilibrium expression. The system will react to the stress by shifting additional reactions forward. So now we will create additional acetate ions. At the end of the day, when we add additional OH ions, the OH ions are not free. They will not be able to distract or disturb the pH reading of the solution. Now, this buffer system will continue to work as long as there are enough of the acetate ions to soak up any hydronium, or conversely, enough of the acetic acid molecules to produce additional hydronium ions. This buffer will fail when we run out of either the acetate ions or the acetic acid. And this is what we call the buffering capacity of the solution, which we'll discuss further on the next slide. So again, the buffering capacity is really the limit of the ability, how much the buffer can handle a change in pH, a change of the acids or bases being added without changing the pH. When the conjugate acid or the conjugate base is completely used up by the addition of strong acids and strong bases, the buffer fails and now the pH will change dramatically. So if we're to look at a graph of the volume of NaOH with a pH, as long as there's enough acetate ions to produce additional hydronium to soak up the OH, such as in this region, the pH will not change dramatically. However, as soon as you run out of acetic acid ions, the pH will now dramatically rise because there is free OH available in the solution to change the pH. Conversely, if we add hydrochloric acid and there is sufficient acetate ions to buffer that change, the pH will not change, will remain relatively stable. As soon as there are no more acetate ions, the pH will decrease because the free hydronium will affect the concentration of hydronium and therefore affecting the pH as well. However, you may ask yourself, which of the conjugate weak acid-base pairs is the best to use to buffer a particular system? Well, it really depends on the pH that you want to buffer. Now, because of our reaction here, buffering capacity is the most effective when it is about equal concentration of your acid as well as your base. That way you can maximize the ability to absorb the excess hydronium or to create additional hydronium. So if we look at our equilibrium expression, when it is most effective, when the concentrations are equal, these terms cancel out, so then the Ka equals hydronium concentration, which means, and then if we log both sides, we have the pKa equals the pH. So therefore, for an ideal buffer, you need to have the pKa, the negative log of the Ka, to be similar to the pH that you want to buffer. You could also add small amounts of a strong acid and strong base to change the ratio of the acid and the base. However, that is beyond the scope of Chem 30, but you may be able to cover that during first-year university courses. Buffers are very, very important in biological reactions, in all biological reactions, because enzymes are the catalyst for biological reactions, and enzymes being proteins, they have a very specific pH value for which they will work. In fact, if you look at some textbooks, you will find that there are often graphs where the y-axis is the reaction rate of that reaction and the x-axis is the pH. You'll often find a very sharp curve where the enzyme is active over a very narrow pH band. And in order to maintain a body pH at a very narrow band, no matter what food or what environmental conditions you are in, the body system requires a very strong and robust buffer system. So the way our human bodies buffer our blood is through the use of the carbonic acid and hydrogen carbonate buffer system. You also recognize this buffer system as the way the body transports carbon dioxide from our tissues to our lungs. This is covered in Bio 20, but we should note that this not only is a way to transport carbon dioxide from the body to our lungs, but it's also critical in maintaining a constant blood pH. Our ideal blood pH is actually a pH of approximately 7.4. If there are plus or minus difference of approximately 0.4 value, the body begins to shut down because enzymes is no longer able to catalyze reactions, such as the reactions required for cell respiration to generate ATP for all cellular processes. Not only are buffers important in our body to maintain our life, the buffers are also important in various commercial processes. So, for example, one is fermentation, the creation of yogurt from milk. Again, this is done by bacteria, and since bacteria are living things, their reactions are catalyzed by enzymes, and we've already talked about how enzymes are only good over a narrow pH range. Similarly, bacteria, not only do they give us yogurt, but they can also generate antibiotics for us. Basically, any reactions where we need enzymes or biological systems, we will need to carefully control the pH. So some of these we discussed already, cheeses, yogurts, sour creams. Some things that are not on the slide would be the fermentation of beer, fermentation of yeast in making of breads, and fermentation of alcohol to make vinegar. Basically any processes that requires enzymes and bacteria to catalyze the reaction.